Today we are going to be talking about Boolean modeling of T-LGL leukemia cell lineages.
Leukemia affects tens of thousands of people around the world. In 2014 alone, there were over 47,000 people diagnosed, half of which died. These numbers are increasing, with over 62,000 expected cases in 2017 and around 25,000 deaths. Large granular lymphocytic leukemia is a rarer form which attacks the lymphocytes, lowering overall red and white blood cell counts. Unfortunately, there is no cure for leukemia. There are just ways to help the patient try and manage. One way this is done is by administering cyclophosphamide in an attempt to suppress cell reproduction. Another way is to treat with cyclosporine and/or methotrexate, which tries to slow down activity of the immune system. Previous studies done by Zhang et al. Have constructed a Boolean model in an attempt to understand the complex signaling components within the pathways of these cancer cells. A Boolean model illustrates different pathways on how proteins reach apoptosis or cell survival depending on the input signal it receives. The output depends on various protein activation or suppression represented by nodes within the network. There is a serious lack of knowledge that lies within the transduction networks of cancer cells that comes from the vast heterogeneity and inconsistency within cancer cell phenotypes. Cancer cells are rapidly mutating and evolving, making each cell unique, imposing serious continuity issues in synthesizing a treatment. Indicating how mutations that are present in cancer cells disrupt or alter gene expression in later downstream processes is highly difficult, especially with the research that shows the difference in genomes from person to person. When treated with various drugs, a large majority of the cancer cells die, but a few remain that have mutated resistance to this specific drug. Looking at how these mutated cells have altered the activity of downstream proteins after multi-round treatments could show how these mutations became so impervious to the drug. However, using the same Boolean model that Zhang et al. Used to construct their survival signaling network, we could see the chain of reactions that occurs when a specific target node is turned on or off within the model to imitate the mutation of the leukemia cells that we observe. These networks could then be compared to how the NFKB protein, which plays a significant role in the apoptosis pathway, is influenced when introduced to an inhibitor such as the BAY11-7082. This could give us insight on how these cancer cells are evading cellular processes like apoptosis, which are essential to prevent cancer proliferation. The research question was, how do the molecules within the signaling transduction network respond to evolutionary forces after multiple rounds of treatment of the BAY signal, and can mutant networks within Boolean model be created after the inhibition of the NFKB protein complex?
After determining the necessary dosages of BAY exposure required for the cell to develop resistance as indicated by continued cell survival, we predict that the Boolean model will demonstrate an output of cell survival even in the presence of an apoptotic signal. This will likely be due to resistance developing in the form of altered downstream protein activity between the Boolean model predictions and the activity present in vitro. By comparing these two, we will alter the Boolean model to match the live cell protein levels, finally resulting in the modeled output of cell survival in the presence of an apoptotic signal.
Our experiment focuses on building Boolean modules of the leukemia cell using a software called Net Synthesis, a tool for building signal transduction network. Each component in the network accounts for a node in the Boolean module, and this node has two discrete states, on or off, depending on its activity level. Here we obtain and simplify a Boolean module of the signaling network for a normal, resting leukemia T cell from previous literature by Zhang. The first step is to obtain the leukemia cancer cells from the consented patient's peripheral blood mononuclear cells. Next, we introduce cells to BAY, the signal that induces apoptosis in normal cells by inhibiting NFKB, a specific downstream node of PI3K. Then we perform adenosine V detection assay, a method to check apoptosis in humans. Based on Zhang's study, we expect to check high apoptosis percentage in these leukemia cells after 12 hours of treatment. We'll conduct functional protein microarrays to check any interaction and activities level of the involved components at 3-hour interval. Based on this result, we then re-update the initial Boolean module nodes, switch on green for active components and off red for inactive components to fit the acquired experimental result after apoptosis introduction. We then reculture the survivors after the first round of PAY treatment. After the regrowth of these survivors, we add another round of PAY for another 12 hours treatment. Similar hypothesis assay and protein array will be performed afterward, so as to alter the Boolean module of the leukemia signaling network. The survivors will again be recultured and constantly exposed to BAE until the cells develop BAE resistance. Anderson assay can help to determine the low percentage of apoptosis in resistant cells. As the cells have developed resistance to the drug BAE, we compare the final updated Boolean module with the resting module to track what specific components, interactions, or pathway have changed as leukemia cells develop drug resistance.
Because BAE signaling directly targets the production of the NFKB protein, it is expected that in our initial trial, its exposure of the drug will result in increases of the NFKB protein activity, and that the cells will die. As the cell develops further resistance with each round of treatment, we expect proteins to be altered from their original pathways. This is because within the pathway, each protein node is either expressed or silenced, resulting in apoptosis. However, as the cell develops resistance, it must change one or more of these proteins. Ultimately, the cell has changed enough of the components of a pathway to result in continued survival even in the presence of an apoptotic signal. Potential mutated proteins that could cause the resistance could be FAS, SFAS, and MCL1 because these could be used as alternate routes to avoid NFKB. Ideally, after comparing the varying levels of protein activity, we will be able to pinpoint particular proteins that have changed, which will allow us to conclude a final pathway through which a cell develops resistance.
Limitations to this experiment are that most cancers develop rapidly, and each time they divide, they have more mutations, allowing them to become death-resistant. In this experiment, it is assumed that the cancer cells will develop a resistance to the drug the cells are being treated with. However, it is possible that when treated with the drug, all the cells will be killed. Also, cancer is a complex disease that has many factors contributing to it. In this case, only T-LGL leukemia cells are being studied. T-LGL leukemia cells have a particular protein network, but in other cancers, the networks may be different. NFKB is the protein being targeted in leukemia cells, but in other cancers, the target protein would have to be different. Lastly, cancer cells are very unstable because of the many mutations, so it is possible the activity of the proteins we are looking at will have varied activity levels. These proteins are important for looking at changes in the protein cascade in the T-LGL cells. If the protein activity is varied, it will be hard to determine if the target protein is reacting to the drug.
Because achieving resistance is paramount to our experiment, our first method for developing resistance is to expose leukemia cells to small enough doses of BAE signaling to allow a portion of the cells to survive and reproduce. To minimize random protein activity levels, control protein levels will be measured in all rounds of treatment. While this computer model is limiting in its ability to measure variation of protein expression because of the required nodes being on or off, the running of this asynchronous model several times will allow us to simulate variability. To ensure that this pathway change occurred in many cells rather than an individual, the protein assays will be conducted for entire cell colonies. Finally, this same experiment could be run with different drugs to model alternate pathways of resistance.
To understand how a cellular pathway works and where the resistance forms. Specifically, in the T-LGL leukemia cells, the NFKB protein was targeted with the BAE signal. This model permits an understanding of where resistance develops on a molecular level. By means of measuring the protein activity levels, this model predicts after how many rounds of treatment the cell will develop resistance, thus an understanding of where inhibition of successful treatment will occur. This model could be used as sequential steps to predict future treatment to select the most promising drug therapy before experimentally searching in live patients. Finally, this model of experimentation could be used with numerous drugs, cancer types, and ultimately could be used to compile a database of cancer signaling pathways.